Hey Team Bio, welcome to your screencast on cell respiration. So just want to clarify quickly, this information is not going to be on your upcoming test, but it will be on the test, a test that you take sometime after April break. Believe it or not, April break is coming up soon, and so this understanding this information is going to set us up to jump right into cell respiration and some exploratory labs in this unit when we get back. So that's why we're doing this now. Okay, photosynthesis. You all know the unbalanced chemical equation by now. We have CO2, we have H2O, we have some light energy, let's just say energy, which isn't really a reactant, but we're going to put it over here because it looks good. And this makes C6H12O6, which is glucose, and oxygen, gas. Okay, so that is the equation for photosynthesis. You can take this equation, sorry, unless you're doing this on an iPad, this might be a little frustrating. You can copy this, place it over here, and copy this, and place it over here. Copy this, place it right here, and change this from light to ATP, and voila, you have the chemical equation for cell respiration. So basically, everything that we did in photosynthesis, which was what words did we use? I think we used endothermic, not endergonic. An endothermic reaction, we had to put in some energy in order to make glucose. Cellular respiration is an exothermic reaction. We're going to split glucose up, and we're going to harvest that energy in the form of ATP energy. They're inverse reactions, and we're going to undo everything that we did in photosynthesis in the process of cellular respiration. Cell respiration is kind of like a controlled bonfire in your body. And about 40% of the energy in this process is transferred from glucose, is transferred to ATP energy. So it's a relatively controlled bonfire, right? A bonfire is just releasing a bunch of heat energy, but cell respiration, 40% of the energy is captured, and then 60% is released as heat. And this is a huge part of why you have you're releasing body heat all the time. If you think about the average car is about 25% efficient, then cellular respiration is actually a pretty efficient process overall. Okay, so this flow chart is, unlike photosynthesis, which actually that's not quite true. We have a couple different forms of photosynthesis that we've been talking about. I'm going to give you a flow chart for the overall movement through the different chemical reactions of cellular respiration in this little graphic right here. Okay, so let's just start it off here. This is cell respiration. All cellular respiration starts with a set of reactions known as glycolysis. Glycolysis, glyco meaning sugar, and lysis meaning to split or to break. So in the process of glycolysis, we are going to be splitting or breaking a specific sugar, glucose. This process occurs in the cytoplasm of the cell. And it creates 2 ATP. When it runs through, on net, 2 ATP are produced. Okay, so this is the first step of cellular respiration. It occurs in the cytoplasm, 2 ATP are produced. It's called glycolysis. Now, we move down our flow chart and notice that there is a fork in the road here. There are two different pathways. One is called the aerobic pathway, and the other is called the anaerobic pathway. The aerobic pathway occurs when the cell is in the presence of oxygen, O2. So if oxygen is around, then we can proceed aerobically. If oxygen is not around, no O2, then we have to proceed anaerobically. There are two different types of anaerobic fermentation that we're going to study in this class. The first type is called alcoholic fermentation. And the other type is called, what color do I want to use? I guess this one, lactic acid fermentation. So these two types of fermentation occur in different types of organisms. So for example, humans, we are lactic acid fermenters. You all have probably heard that if you're sprinting, you're performing anaerobic work, and you are producing lactic acid. The same thing is true in your refrigerator. The bacteria that are in your yogurt are lactic acid fermenters, and that's what gives yogurt its kind of tart, sour taste, is in part the lactic acid that they're producing. Yeast, on the other hand, are alcoholic fermenters. If you leave yeast in a sugar water solution, they will perform alcoholic fermentation, and ethanol will be produced. So, depending on what type of organism you are, you're either doing alcoholic fermentation or you're doing lactic acid fermentation. Humans do not do alcoholic fermentation. So that would, if you were sprinting at practice, you would be getting drunk. But that's not what we do. We do lactic acid. So organisms are either this or this, but not both. There might be an exception to that rule, but for all I know, that's true. Okay. So both alcoholic and lactic acid fermentation occur in the cytoplasm, and they are happening when oxygen is not present. And we're going to talk about what these processes do later on. Okay, if oxygen is present, we can burn our glucose much more efficiently. So this is where the products of glycolysis proceed to a set of reactions called the prep steps. And the prep steps, along with everything subsequent to the prep steps, happen in the mitochondria. Remember, the mitochondria is the powerhouse of the cell, and you'll see why in just a minute. Okay, following the prep steps, there is a set of reactions called the Krebs cycle. You'll notice when we say the Krebs cycle, it bears a lot of similarities to the light independent reactions, except kind of moving in the opposite way. Instead of putting high energy electrons on a carbon molecule, we're going to be stripping them away from a carbon organic compound. Okay, and then I should say the Krebs cycle produces 2 ATP. And then the last part of the Krebs cycle, of the aerobic pathway, is called electron transport phosphorylation. And you all now know this word, phosphorylation. You know this is when a phosphate group is added to something. And yes, here a phosphate group is added to ADP to make ATP. And this process creates somewhere between 32 to 34 ATP. And we'll be talking about this discrepancy, why we don't have an exact number here, in a little bit, or later on in this process. Okay, so that is cell respiration. Not the nitty-gritty details, but just the overall flow through it. It always starts with glycolysis, which occurs in the cytoplasm. If oxygen is not present, then an organism is either going to do alcoholic or lactic acid fermentation. And if oxygen is present, we will undergo the Krebs cycle and electron transport phosphorylation. And the whole point of this darn process is to produce ATP, as you can see, produced here, here, and here. Okay, so last up, we're going to talk about the structure of the mitochondria, because this is where an important amount of the reactions in cellular respiration take place. Okay, the mitochondria is an organelle, but you all know it is the result of an endosymbiotic event in which an ancient bacteria was engulfed by an ancient archaea, and that gave rise to all modern-day eukaryotic organisms. The mitochondria was that little bacteria that didn't get eaten and stayed on living mutualistically inside that archaea, gave rise to the modern-day mitochondria. Some of the relics of that ancient event is that the mitochondria contains some of its own DNA and some of its own ribosomes. Some of the genes that the mitochondria, the proteins, the genes for the proteins that reside in the mitochondria are made from DNA in the nucleus of the cell, but some of the mitochondrial DNA resides directly in the mitochondria. The mitochondria is composed of two lipid bilayers. The outer one is surprisingly called the outer membrane. And the inner one is called the inner membrane. The space between the outer and inner membrane is called the intermembrane space. And then the space that's within the inner membrane has a fun name. It's called the matrix. The mitochondrial matrix. Okay, you'll notice that the membrane, the inner membrane is highly folded. This is, yes, to maximize surface area to volume ratio. The important reactions are going to be happening on this membrane, and so by folding it up, you can get a lot more of it inside the same amount of space. These folds are called cristae. And on the inner membrane are some things that are going to look familiar to you. We have electron transport systems, and we have the membrane protein that we all know and love, ATP synthase. Okay. And that is the basic structure of the mitochondria. We're going to be talking more about especially how these steps relate to this structure, where they're occurring, and how. Okay, but that's all for today.